Thanks, Suchun, for the introduction. And we have been waiting for this symposium in Singapore for too long, probably 10 years. And it's my great pleasure to have the honor to speak our work in the very first talk. It's very exciting. So thanks for the organizers, especially Adam, Suresh, AJ, Fuyu. So I'm not naming all, but thanks really all of you for your effort for making this happening. So my lab is interested in understanding how the transition of quiescent neural stem cells to become reactivated. So my lab in the history has been working on understanding how, so all kinds of interesting behaviors of neural stem cells, how do they divide asymmetrically, how do they undergo self-renewal and differentiation. And this is a relatively recent direction in the lab, and I'm really fascinated about. So in the adult mammalian brain, most of the neural stem cells are actually present in a mitotically dormant quiescent state. But in the presence of stimuli, they can be re-entering the cell cycle and become active again to generate new neurons. So lack of the regeneration capacity in the mammalian adult brain can be attributed to one of the reason that the lack of regeneration capacity could be this not many actively dividing stem cells around ready for repair and regeneration. So the ultimate goal is that I hope our study can eventually help to generate new knowledge on how to stimulate the endogenous neural stem cells and use them as a therapy to stimulate our own endogenous neural stem cells for regeneration capacity to generate new neurons and compensation of neuronal loss in neurodegenerative disorders. So studies in mammalian brains has also shown that the impaired neural stem cell reactivation can lead to a deficit in adult neurogenesis and decline of cognitive function. But yet we know very little about how to wake up the neural stem cells, quiescent neural stem cells. So we are using Drosophila model, and there are certain advantages. First of all, they all relatively undergo synchronized quiescence, enter quiescence at the end of embryogenesis. Within about 24 hours, they all actually be reactivated also in a relatively synchronized way. So that means if you look at the first few hours in the larval life, pretty much all of them are in quiescence. And if you look at beyond 48 hours, all of them are proliferating. So this makes the system really amenable. So it's also very easy to distinguish the quiescent neural stem cells from the reactivated ones because they extend a cellular extension projecting into the neural pils where there's a lot of axon bundles and neural pil glia. So these are the reactivated ones. So invariably, all the quiescent neural stem cells, before they become reactivated, they will retract this cellular extension, cells become rounded up, they then undergo cell growth, and then they can divide asymmetrically to generate the cells, smaller cells surrounding them eventually give rise to neuron. So how to wake them up? So a lot of studies shows that the Prince Charming actually turns out to be the essential amino acids in the food. So this is dependent on this series of signaling relay from fat body, which is an invertebrate, adipose tissue, and liver, and then transducing signal to blood-brain barrier glial cells, which then secrete insulin-like peptides, activating PI3K and AKT pathway in the underlying neural stem cells. So basically, the insulin pathway promotes neural stem cell reactivation. And another pathway called HIPA pathway actually maintains the cells in quiescence. So there's a lot of ongoing battle over that, when and how to activate the neural stem cells. So now all this work will actually originate from a very unexpected finding. About 10 years ago, we were doing RNA screen, and an undergrad student told me that he found this weird neural stem cells with a tail hanging behind. So at the time, we didn't know what's really going on. We have never seen this phenotype before. And so through many years study, we now understand that these neural stem cells stay in quiescence for the entire larval life. So basically, they stay in quiescence, and then our work 2 years ago showed that this is an intrinsic mechanism regulating neural stem cell reactivation by controlling gene expression. So today I would like to focus on E3 ubiquitin ligase named CR4. This is a protein complex that it play a pleiotropic function in cell cycle and proliferation. So it's basically the E3 ubiquitin ligase targets substrates for ubiquitination and subsequent degradation by proteasomes. So this complex is composed of several core subunits, the scaffold subunit calin-4, the adapter protein DDB1, and the ROC. And DCAP is a variable component, DDB1 calin-4 associate factor. So it binds to substrates and provides substrate binding capacity. So very interestingly, variants in human calin-4 are associated with mental retardation and malformation of cortical development. But whether calin-4 actually plays a role in neurostem proliferation was poorly understood. So we isolated loss of function alleles of DDB1, which is a core component of this calin-4 complex. So you can see that they formed really tiny brains, so we call it microcephaly-like phenotype. So you can see that from the quantification, there's dramatic reduction of the brain volume. So this is likely due to proliferation defects, and indeed, we look at two things, basically the EDU incorporation, and look at whether whether they're able to undergo in cell cycle, and we also look at how many cells still retain the cellular extension, which is now considered to be a hallmark of quiescent neurostem cells. So as you can see that there's a dramatic decrease of cells incorporating EDU, suggesting that they are not cycling. And there are also a dramatic increase of cells retaining the cellular extension, suggesting that they are staying in quiescence. So DDB1 is important for neurostem cell reactivation. So we look at another core component named calin-4. So calin-4 loss of function allele also showed even stronger phenotype. So you can see that there's very dramatic decrease of EDU incorporation, and there's very dramatic increase of cells still keeping the cellular extension, meaning that they are still in quiescence. So what about the DCAP protein, which recognizes substrates? So there are around 50 genes, 50 DCAP genes in fly genome, and we carried out an RNA screen to identify which DCAP protein is important for reactivation. So we found a single DCAP protein called Mahjong in Drosophila, or DCAP1 in mammalian cells. And Mahjong was previously shown to regulate cell-cell competition, and the name was interesting because so basically the Japanese postdoc and Chinese PI agree that this is the best name for it, mimicking cell-cell competition. So as you can see that the RNA of Mahjong leads to reactivation defects. So if you look at the homozygous mutant, this again very much like a microcephaly-like phenotype. There's a dramatic decrease of EDU incorporation and dramatic increase of cells retaining the cellular extension. So very interestingly, when we overexpress Mahjong, we can observe the opposite phenotype. So at very early stages of at the larval life, so at 6 hours, majority of the control are not proliferating. So this is EDU positive cells. And as you can see that when you overexpress Mahjong, you can see a majority of them are not proliferating. So that means overexpression Mahjong can drive your stem cells into premature reactivation. So what is the target of the CR4 E3 ligase in neuroblast reactivation? So we thought that the Hippo pathway would be a good candidate, as Hippo pathway keep the neurostem cells in quiescence. So during the neurostem cell quiescence, the Hippo pathway will be activated, and the depletion of the Hippo pathway components will result in a premature neurostem cell reactivation, and this is very similar to Mahjong overexpression phenotype. So we propose that the CR4 E3 ligase targets WOTS for ubiquitination degradation. So we first showed that the Mahjong and WOTS form a protein complex in the co-immunoprecipitation experiments. And this interaction also facilitates the association between DDB1 or calin-4 with WOTS. So basically, they are associating with WOTS in the presence of excess Mahjong. So we then overexpressed Mahjong and did a ubiquitination assay. We found that this is able to significantly enhance the ubiquitination of warts. In contrast, the N-terminal Mahjong lacking the substrate binding domain is unable to enhance the ubiquitination. So basically, this data suggests that Calin-4 promotes the ubiquitination of warts. So because there's no warts antibody that is available to detect the protein level or works for immunostaining, so we decided to look at use Yoki as a readout. So basically, in the loss of function of a CR4, we would expect accumulation of warts, and this would result in phosphorylation of Yoki, and then resulting in Yoki's cytoplasmic retention. So indeed, in the wild type, at 24 hours after larva hatching, most of the Yoki are localized to the nucleus, co-localizing with DEPN, which is a neurostem cell marker here in blue. So in the loss of function of CR4 mutants, Yoki is largely cytoplasmic or undetectable, suggesting that they are inactive. So all this suggests that support our model that CR4 targets warts for polyubiquitination and then degradation. This will release Yoki into the nucleus and therefore triggering neurostem cell reactivation. So what is the next? So we'd like to take this further to the mammalian system to look at the function of CR4 E3 ligase in mouse and human neurostem cell proliferation. So we would like to generate human cerebral organoids model to understand whether CR4 E3 ligase is important for the similarly important have a role in neurostem cell proliferation in the mammalian system. So we are able to grow these organoids, and we are able to see the different layers in structures in the organoids mimicking the human brain development. So I hope you can appreciate that this is a giant leap, hopefully, from Drosophila to human, and then we are pretty excited to test the function in the higher model organisms. So the CR4 work in Drosophila is mainly done by a very talented PhD student, ex-PhD student Tao, and the mammalian work is really spreadsheet by very talented postdoc Dong Liang. So thank you all.
Any question, comments? Maybe you can move to the microphone if you have any question. We can still have time for a couple of questions. I thought that the CIL4 is regulating the ubiquitination, right? Is that right?
Yes.
It could regulate a lot of things like cell cycle proteins and so on and so forth, because that's what we also found that does it regulate like a cycling, all sorts of protein in your system? And so that it may affect the cell division or something like that.
Yeah, it does. I mean, it does regulate a lot of cell cycle regulators, including cycling. And so we think that the Mahjong has more specific function compared to the Calin-4 and DDP1 because there are at least 50 different decap proteins, and we only found one, which is decap1 Mahjong, that has the function. So it means that Calin-4 and DDP1 can have really pleiotropic function targeting different substrate, targets. But through decap1, it has more specific targets like Hippo pathway. Yeah. So I mean, basically, we do see that, for example, in addition to the neurostem cell proliferation, reactivation defects, DDP1 and Calin-4 also have other additional defects in like regulating asymmetric cell division at later stages, and this is not true for Mahjong. So we think, but in vivo, there's a lot of different Calin-4 complexes by through different decap proteins and different targets. So I think through Mahjong is relatively specific.
Just one simple question. It's really nice to see you're translating your findings in Drosophila into mouse and humans. For that protein CRL4, it is very well conserved from Drosophila all the way to human.
Yeah.
And
So is that, I mean, for example, Calin4 has 2 homologs in humans, Calin4A and B. So 4B has been reported to be, mutation associated with neurodevelopmental disorders. So that has been very well reported. I mean, there's a lot of studies that linking to mental retardation and different kind of neurodevelopmental disorders. And Mahjong, which also has a human homolog called DCAP1 in human as well.
very nice.
Adam.
Hi, Hongyan. Yeah, nice talk. If you're able to, say, reproduce the results in the mammalian system, what would your strategy be to convert these findings into a therapy? Like, what would the overarching strategy be? I mean, I know that's a very open-ended question, but
Yeah, so
in general terms.
okay, so I think in the limited timeline, what I've been thinking about, for example, Mahjong overexpression is able to push the cells into earlier reactivation. So of course, it remains to be tested in the long term whether that's an advantage of producing additional neurons or not, right? So we hope that we are able to find a condition that we are able to drive, additional neuron formation without causing tumors.
Okay, maybe if you can speak loudly.
Yeah, yes, yeah, the reviewer definitely will ask. So in Drosophila,
Sorry, Hongyan, could
especially,
you repeat the question?
Oh yeah, the question will be that for our mutants or knockouts, have we carried out rescue experiments? So my answer is yes. So as a Drosophila geneticist, we are very solid on that. So every mutant loss of function alleles has been rescued by putting back the wild type version. Yeah. Yeah, so the Mahjong has been shown to be localizing both the nucleus and the cytoplasm. And so we don't know, I mean, I think the DDB1 complex also, the DDB1 also itself has also been shown to translocate into the nucleus in response to DNA damage. But the translocation to the nucleus may reflect additional function other than the reactivation. So we think the